Hello dear students, today we are going to deal with the antiprotozoal agent. This is the first part of the antiprotozoal agent. Myself Arif Khan, Senior Lecturer, Institute of Pharmacy, Diploma Azim Campus, Camp Pune. We are dealing with the antiamoebic agents. So antiamoebic drugs, actually antiamoebic drugs are the drugs used in the treatment of amoebiasis. So, amoebiasis is nothing but what? Amoebiasis is a parasitic protozoal infection caused by Antiamoeba histolica. So, Antiamoeba histolica is one of the protozoa, and these all are the microscopic images of these Antiamoeba histolica, all three including. This is the tropozoid stage. So, this is actually the Antiamoeba histolica, and the disease caused by Antiamoeba histolica is called as amoebiasis. The disease is due to the poor hygienic conditions and is transmitted by the fecal oral route. So definitely this the route mode of transmission for this disease is fecal oral route. Amoeba are present in particularly the lumen or a wall of the gut of the human beings, and these will able to cause the infection. So basic site for presence of the amoeba or Antiamoeba histolica will be the lumen of the gut or the intestine, and they are being enlarging over there. These are some of the stage like encystation, then cyst formation and excistation, and then after that these a grown stage that we call tropozoids. This will show the microscopic structure of the tropozoid. It look likes okay, and these are enlarging on the particularly on the lumen side, particularly on the villi as well as on microvilli. So these are get replicate themselves and they are able to spread the infection. Now next one, as far as the chemical classification of these antiamoebic agents are concerned, in that first is alkaloid, example imitine, dihydroimitine. Next amaroid, the example is a causine. Next antibiotic, paramomycin is an antibiotic which is used as an antiamoebic. Quinoline derivative, and that halogenated hydroxyquinoline, that diiodohydroxyquinoline is called as idoquinol, and idochlorohydroxyquinoline is called as cleoquinol. Next is the aminoquinoline derivative, in these chloroquine and amodiaquine. These are the examples. Next class for these anti-amoebic agent, that is heterocyclic compounds containing furan derivative. Furan will be heterocycle in these particular drug. Diloxanide furorate, it is an example. Next, imidazole containing imidazole derivative, metronidazole. Next, the organometallic compound, which has organometals in their structure like acetarsol, that is arsenic containing compound. Arsenic is a heavy metal or organometal. So acetarsol, it is an example of the drug which is used as anti-amoebic. Now if you're talking about the exact location of the lumen or where which these tropozoids or these amoeba, Antiamoeba histolica get enlarged and survive, so it will be in the intestine. So say we are talking about this is a intestine, small intestine, which this is internal layer or internal mucosal layer we are calling a lumen. Okay, internal part or internal layer is called as lumen. Now this lumen, if you are seeing the TS, so it will have the number of villi. So this we are taking expanding this part. So this will be the villi. So number of villi will be present, and on each of villi we can see that this can be enlarged to this one. These are the microvilli. Okay, these are the microvilli. So on the lumen there will be the number of villi, and on the villi there will be a number of microvilli, and that thereby that will be increasing the surface area, and this reddish part indicate this is the lumen, which may be the part of villi or microvilli, and on this part particularly these Antiamoeba histolica are enlarging, surviving and multiplying, particularly in the tropozoid stage, may forming a C stage for this spreading to the fecal matter, and will get enter into the new person through the fecal oral route. So that is the mode of transmission of the Antiamoeba histolica. Next way of classification of the antiamoebic agent, that is on the basis of their site of action. Those acting on the lumen called as luminal antiamoebocytes. These act mainly on the bowel of the lumen. Diloxanide furorate and parabomycin. These are the example of the drug which are acting as these luminal amoebocytes and halogenated hydroxyquinoline. Next, tissue or systemic amoebocytes. So particularly these first one will be acting as a local, and second one will be acting as tissue or systemic amoebocidal agent, which will be acting systemically. So these are the drugs which are act on the wall of the intestine and the tissues. So this include the Imitine and its derivative, and mixed amoebocytes which will acting by both the way. These act on all sides of infection such as intestinal lumen and the intestinal wall and the other tissues. So that's why majorly the classification of the amoebiasis will be as symptomatic and asymptomatic. Wherever there is a symptomatic, so that will show the luminal amoebocytes and the extraluminal amoebocytes, extraluminal amoebiasis actually. So luminal amoebiasis will limited to lumen only, and extraluminal amoebiasis can invade into the other tissues also. So these particularly mixed amoebocidal agent will will be effective in both the types of infection, both the types of amoebiasis, that is luminal and extraluminal amoebiasis. So for that we have example metronidazole. Irrespective to the type of these luminal or tissue, this will be act. That's why this is called as mixed amoebocytes. Next, now we are moving towards the drugs. In that first, that is the Imitine. So Imitine, as far as the physical properties of Imitine is concerned, it is a white to slight yellow color, odorless, beta crystalline powder, and it is soluble in water. Now as far as the chemical property, it gives the bright green color when mixed with the molybdenum trioxide and sulfuric acid sprinkle on it. Next chemical property, it contains the two basic nitrogen atoms, hence it forms the salt quite rapidly with the two molecules of hydrochloric acid, and it will give the Imitine hydrochloride. So it is basic in nature, it will form salt with the acid, that is HCl with the HCl, and it will form hydrochloride salt. So next, stability storage of the Imitine, it is stored in tightly closed light resistance container. As far as the uses are concerned, it is the tissue amoebocytes, and hence it is used in the treatment of intestinal amoebiasis, then hepatic amoebiasis, which is then say extraluminal. Next, amoebic infection of the lungs, brain, and other tissues. Again, this will be again the part of extraluminal amoebiasis. So in both the type of amoebiasis, the Imitine will be effective one. Pharmaceutical preparations include the Imitine injection, and the brand name include Stemibin and Hemomatin. These are the brands for this drug Imitine. Next, metronidazole. Now as far as the chemical name of this metronidazole is concerned, now the basic nucleus is actually imidazole in these, or we can say that basically we are taking parent nucleus as ethanol. So if we are giving say numbering to this ethanol, so we'll give this as far as functional group, hydroxy group is the functional group in this. So this carbon will be the first carbon and this carbon will be the second carbon. Now on the second carbon, this is a say complicated substituent which is called as imidazole 4-il, or we will give the say for example if we are giving numbering, or this is as a complicated substituent, separate numbering if we are giving to this imidazole, this will be 1, this will be 2, then 3, then 4 and 5. So you can see that on ethanol, second position of the ethanol, there will be say 2-methyl 5-nitroimidazole 1-il. Okay, 1-il. So this will be the say we can say that on the ethanol on second position. So ethanol we are writing at the end name of the parent nucleus, and next one that is the as far as the say complicated substituent is concerned, so it will be in a bracket, that is 2-methyl 5-nitroimidazole 1-il, that is 2-methyl 5-nitroimidazole 1-il, because it is attached, imidazole is attached with the first position to this parent nucleus ethanol, that is why 1-il. So 2, this is the position of the ethanol. On second position of ethanol there will be the 2-methyl 5-nitroimidazole 1-il, this complicated substituent which we have included in the bracket, and ethanol is a parent nucleus of the given structure, is called as metronidazole. So this is the one of the drug which has a nitro group in its structure, that's why nitro coming, that's why metronidazole, this is the say trial name for the given drug. Next, as far as the chemistry is concerned, it has a 5-nitroimidazole derivative. It contains 2-hydroxyethyl group and the methyl group on the 1 and the 2 position respectively. Oxidation either of these group leads to inactivation of the compound. Next, physical properties, that it is a white or creamy white color, slight odor, bitter taste, and crystalline powder and slightly soluble in water. As far as the chemical properties, when its solution in sulfuric acid is treated with the picric acid, it gets precipitated as a picrite salt, which melt at 150 degrees Celsius. Then nitro group of the metronidazole on reduction with Zn and HCl, it will convert into the primary amino group, and which will undergo the dissociation reaction with the HNO2, that is sodium nitrate, and the HCl. So as far as this is a chemical property of this metronidazole. So as we already discussed the structure, that in which there is a nitro group, and that's why metronidazole. So nitro group first to be reduced with the Zn and HCl. So it undergo the reduction, it converted into the amino group, and we are calling it a primary amino group. And if there is a formation of primary amino group, now the primary amino group is available for the diazotization reaction. So for this diazotization reaction, the sodium nitrate and HCl is used as a reagent at the temperature condition 0 to 10 degrees Celsius, and this primary amino group undergo the diazotization reaction. So this is the way. First we have to say reduce this NO2 group to primary amino, then primary amino undergo the diazotization reaction. Now the diazotized product can be coupling with the beta-naphthol in NaOH and gives the orange red color. This will be the confirmatory test to identify that whether this diazotized product has been formed or not. So for that, this was the identification test or the chemical test. Next, metronidazole stability and storage. So it should be stored in tightly closed light resistance container. It is used in the treatment of the amoebic dysentery, amoebic hepatitis, and the liver abscess by the Antiamoeba histolytica. I mean simply an extraluminal amoebiasis, amoebic dysentery, this is a luminal one, or amoebic hepatitis, it's again I'll say extraluminal amoebiasis. So in this treatment of these, one can use as an anti-amoebic agent metronidazole most preferably. In the treatment of giardiasis, that is diarrhea and dysentery symptoms due to Giardia lamblia. So in the treatment of giardiasis also, these metronidazole can be used. Then genitourinary tract infection. In the treatment of genitourinary tract infection, and like in the treatment of septicemia, pneumonia, post-operative wound infections due to an bactericidal action. So for this also, these metronidazole is effective one. Next, in the treatment of ballontidiasis, that is diarrhea due to the Ballontidium coli. Now pharmaceutical formulations: metronidazole tablet, metronidazole injection, metronidazole suppositories. So these all are the formulations of metronidazole. Now the brands. So as far as the brand names of these metronidazole: Flagyl, Metroquine, and Metroquinol. These are the brands available in the market. Amongst these, the Flagyl is the most popular brand, which may be say popular for the metronidazole or its combination. Okay, so next types of antiamoebic agents we will see in the next part. Thanks for watching. Thanks for being with us.